Hello, Biology 30. Hope you're all doing well. I thought I would show you an example of how to do transcription and translation protein synthesis practice worksheets where you're having to transcribe DNA into mRNA and then translate it into tRNA and then come up with amino acid sequence. Will show you how to do the one worksheet that's DNA RNA practice or transcription translation practice. In order to do this, all you need to know is your little code here and complementary base pairing. When you're given DNA and DNA is A, the complementary base pairing for A or adenine, when you're going to RNA is uracil. If it was DNA, it would be thymine. Now, if DNA is thymine, when its complementary base pairing, when you're transcribing into RNA, is adenine. Cytosine's complementary base pairing is guanine, and guanine is always cytosine. So students don't usually have trouble with the C's and the G's because basically every time you see a C, the next line you write a G, or anytime you see a G, you write a C. Where students get confused is with these thymine and uracil, because if it's DNA, adenine's complementary base pairing is thymine. If it's RNA, adenine's complementary base pairing is uracil. So these T's and U's can sometimes confuse students. So let's take a look at our first example here. This is similar to the worksheet I sent you in your package. So I've given a DNA sequence here, template that I have to transcribe into mRNA. So all I'm going to do is remember my complementary base pairing to do that. Adenine's complementary base pairing is uracil because this is mRNA. If it was a DNA, if I was just trying to do the other strand of DNA, it would be thymine, but this is RNA, so it's uracil. Thymine's complementary base pairing is A, G is C. C is G, G again, C, C, so G, G, G is C. C is G, A is, now don't be tempted to put a T here, you have to put a U because this is mRNA, and T is A. T is A, G is C, oops, that should be a C. I make that mistake a lot. And G is C. All right, so now you're going to do translation. You're going to figure out what the tRNA anticodon letter combinations would be. And so you're just doing your complementary base pairing again. U's complementary base pairing is A. Adenine's complementary base pairing is U because this is RNA. C is G. G is, oops, I did it again. C, 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 G. G is C, A, U. A is U, C, C, so this would be G, G. All right, now all I have to do is figure out your amino acid sequence. And to do that, you're going to use the little pinwheel here. I find the pinwheel is easier than the chart above, but you can really do whichever one you want. The pinwheel is also found on page 367 in your textbook. Now, when you're using this pinwheel, it's important that you remember that the pinwheel is based on mRNA, the codon sequences. Lots of students read the wrong line when they're using the pinwheel, and they'll look at tRNA and they'll look at all the wrong amino acids. So you don't want to do that. You want to look at mRNA. It's a codon chart, so you need your mRNA. So for an example, my first mRNA codon is UAC. So if I go back over here and I look, my first base is U, my second base is A, my third base is C, so my amino acid here is tyrosine. Now we usually don't write down the entire word because it's too long and who wants to write all that? We usually just write the first three letters. It makes it a lot easier. So that's your first amino acid. My next one, if I read the mRNA, it's G, G, G. So I go back to my pinwheel and I look, G, 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 I get glycine. And so here is glycine. And I just keep doing that for the remainder of my polypeptide chain. And then when I'm done, I would have my protein. Now, example 2 is similar, except I kind of made it a little more interesting in my opinion because I mixed it up a little bit. Sometimes I gave you mRNA, sometimes I gave you tRNA, sometimes all I gave you was an amino acid. And you have to try and fill in all the blanks, just remembering your code. So let's start with the first one. I gave you mRNA, so it's CCC. So mRNA will transcribe into G for DNA. And it would also be G for tRNA. And then I look up CCC on my pinwheel. CCC, and it's proline. Now, with this one, I gave you tRNA, ACC. So I need to figure out my mRNA and my DNA and my amino acid. So A, because I'm dealing with RNA, will transcribe or complement to U. C is G. C is G. Then when I do my DNA, U will transcribe to A. G is C. G is C. Whoops. There we go. All right, and then I look at my amino acid, UGG. I go over here, UGG, tryptophan. That's the amino acid that apparently there's a lot of it in turkey. Okay, now this last one is the trickiest one because all I gave you is the amino acid sequence, lysine. So I go over here and I look for lysine. And where do I have lysine? It's right down here. Now there's more than one codon chart that I can use, message. I could go AAG or I could go AAA. Doesn't matter which one you choose, you get to have a little bit of choice here. It's totally up to you. So I'm going to use AAA, and remember that is mRNA. So I'm putting it in my mRNA column. Now my DNA, remember this is DNA. So A's complementary base pairing in DNA is not uracil, it's thymine. So it'd be T, T, T up here, and my tRNA would be U, 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 and then I'd be done. And that's how you do complementary base pairing. One thing I want to point out is your DNA and your tRNA lines, they should be exactly the same. The only difference is T's and U's. Other than that, they're the same. G, 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 G. All right, that's it for now. Bye.